Let me just share my screen.
Good afternoon.
Uh, Hello, everybody. I hope my screen is visible, and I'll shortly begin with the session as well. This is another one of the practice sessions for this course, that is cell biology. I am Arveen Kaur. I'm a PhD scholar at Department of Biological Sciences and Bioengineering at IIT Kanpur. And along with this, I'm also a PMRF scholar. NPTEL has given us the opportunity to kind of conduct these practice sessions for you guys for multiple NPTEL courses that are running currently. So I am a TA for this course that is being taken by Professor Shikha Laloria. I'm sure you are going through the lectures that are being uploaded, the lectures by ma'am and some of the other professors from all around the world. And I hope that you are having a good time with the course and you are learning a lot about cell biology. Uh, so what we'll do in this session is that it will be divided into two, major segments. In the first segment, what we'll do is we'll go through the previous year's assignment pertaining to week 7 lectures. Right. So, whatever we discuss, we'll also discuss the rationale behind the answers, the concepts that are linked to those answers, etc. In the segment two, we'll discuss about some extra concepts. So, the concept that I have picked up for today's lecture is the one that I left, at a cliffhanger in the previous session, the Lambrusch chromosomes and the different characteristic kind of chromosomes that are there, which have given us insights into how chromosomal packaging or how chromosomal structure might be packaged even in higher eukaryotes and mammals themselves, right? And so we look a little into that. Before that, let us just begin with our previous year's assignment for this week 7. So I'll be presenting the first question. Yeah. So the first question for this particular assignment is pick the correct statement about senescence. Option A is senescent cells remain irreversibly arrested at G0 phase. B is senescence is an active cellular death process. C is it occurs only due to infection. D is chromosome fragmentation and cell shrinkage are main characteristics. And E is all of the above. So I would like you all to go through all the options and then we'll discuss the answer.
Option A, ma'am.
Option A, yeah, that is actually the correct answer. That in fact, the correct statement over here about senescence is this one, that is senescent cells remain irreversibly arrested at G0 phase. Right. So what are senescent cells? These are basically cells that the definition of senescent cells is this that they have cell cycle arrest, right? They are usually a product of or a result of aging, normal physiological aging, right? And it is not an active cellular death process, right? What is an active cellular death process? It is apoptosis, right? I hope you did recall that. It does not only occur due to infection because there are multiple, normal cellular aging processes like when cell is aging and dividing over years and years of time. It will accumulate a lot of reactive oxygen species, reactive oxygen species, and other cellular stress proteins and markers, which will eventually age the cell in this process, right? So it will not just be due to infection that senescence occurs. Okay. Then chromosome fragmentation and cell shrinkage are the main characteristics. No, this is also not true. Now, this is another feature of apoptosis and not senescence, right? And since these three aren't true, all of the above is out of question over here, right? So over here, I have also shown a representation of the normal cell versus a senescent cell. So you can see that the normal cell has a very, custom morphology that we are used to looking at, right? So it has all the cellular compartments in place, right? The proportion and size of these cellular compartments is also correct. When they age over time and there is stress that accumulates, stress granules that accumulate, and there is some kind of damage that the cell, like every time a damage occurs, it will try to repair that damage, right? But over time, some amount of damage will still start accumulating, right, as the cell is aging. So it will slip and it will not be that effective in repairing damage, right? So these kind of conditions will lead to the cell's morphology changing a little, right? Along with this, it will be arrested. There will be no progression of cell cycle now. It will be in a quiescent stage, right? And it is a senescent cell. Over here, you can see that most of the cell organelles are still in the same proportion, right? So there is no change that is occurring to the cell organelles. The nucleus is also the same. Within and outside the nucleus, it looks the same. There is just some kind of, overall physical morphology of the cell is changing a little, and along with that there is one other chemical characteristic of these kinds of cells, that they secrete a very characteristic set of proteins which are known as senescent secretome, right? So this senescent secretome is basically one of the features of a senescent cell, right? So how will you identify senescence? So if it is not visible under a microscope, if it is not differentiable under a microscope because of its morphology, right? How else can we check? We can basically we'll know what are the characteristic proteins or characteristic kind of markers that are present in this senescent secretome, and we will design probes to label these, right? If we see that the cells contain in and around them these markers, we can very nicely conclude that okay, this is a senescent cell, it is not a normal cell, right? Because in case of a normal cell, this would not have been the case. These markers would not have lit up, right? Okay, so moving to question number two. Question number two is, which of the following is not a hallmark of aging, right? Uh, Option A is telomere attrition, B is mitochondrial dysfunction, C is stem cell exhaustion, and D is low ROS. So, this is a question from the 9 hallmarks of aging that were given by, I don't remember the name of the group, right? Ma'am has mentioned it in her lecture. And in, I think, a paper in Cell in 2013, these hallmarks were described, and the same group has recently published a revision of the same hallmarks, and now there are 13 instead of 9. But for now, we'll just keep these 9 in mind. And the answer for this particular question is low ROS. Okay, sorry, I couldn't change my slide. Yeah, low ROS, right? Low reactive oxygen species. Now, before I explain this, let me just briefly, tell this to you guys if you don't already have this in your mind. So, what is reactive oxygen species? Right? Reactive oxygen species are the the nascent kind of oxygen species, right? So, it could be a superoxide, single oxide, H2O2 species, right? Which are kind of toxic for the cell, right? If there will be a normal condition under that, the ROS levels will be less in our body, right? So that is why we have a lot of healthy kind of diet that is being promoted. Like there are advertisements that, okay, this green tea is antioxidant in nature, right? So what will that antioxidant do? It will basically go and scavenge these free oxygen or reactive oxygen, and it will quench them, right? So that they cannot stay around in the cell and show their toxicity, right? So that is why a lot of, so it is a general knowledge now, right? It is not just pertaining to science that you should know this, but in general you should be aware that reactive oxygen species is bad for us, right? It will somehow create some kind of toxicity in our cell, right? The mechanism being understood is the biology, but this being known is more of a general knowledge at this point, right? So, because there are a lot of brands that are utilizing this knowledge to basically, uh, make a selling point out of their products, right? So, yeah. So, what was I saying? Yeah. So, low ROS is good, right? So, it will not be a hallmark of aging. Aging will occur when there are multiple other features, among which when the reactive oxygen species inside the cell is going up, right? So this one is definitely the answer over here. Along with this, there are these other hallmarks of aging, right? They are divided in this particular representation into three major categories. One are the primary hallmarks, right? The primary hallmarks or the cause of the damage, as it is listed over here, is genomic instability, that while dividing or while undergoing these regular replication processes, it might happen that now, as we age, our protein machinery and our cellular machineries in general, or replicative machineries or polymerases, are not as well in their function, or not as apt or apt to perform their function as they were when we were younger, maybe, right? So these they might lead to more error prone replication or error prone divisions, right, which might cause genomic instability, right? It could also occur due to other environmental factors inside and outside the cell, right? So some environmental factors, for example, could be the presence of ROS itself or other stress markers, right? Then telomere attrition is another cause of the damage. What is telomere attrition? What are telomeres? Telomeres are these repeated sequences that are present on the end of the chromosomes, right? These kind of protect the actual genetic information that is present in the chromosomes, because it is considered that over time with aging, it might happen that the ends of the chromosome are being pruned continuously, or they shorten continuously, right? A little bit after maybe a few divisions or with each division, right? They keep on shortening, right? So the shortening of these telomeres, if it keeps on occurring, it will eventually affect the genes also that are present on the ends, right? So telomere shortening is associated with increased aging. Right, so that is what telomere attrition is. Then is epigenetic alterations. Now, epigenetic alterations might be alterations not in the sequence of the DNA or sequence of the gene itself, but in maybe its methylation pattern, or its acetylation, or some kind of histone modification, or some kind of a, structural modification in it, such that maybe the normal enzymes or the normal transcription factors that come and bind to it for transcription to be able to occur are maybe now that location where this transcription factor would bind is now occupied by some acetylation site, right? So now this transcription factor is hindered from coming and binding on this genome, right? So now transcription cannot progress. So that kind of changes will be epigenetic alterations, wherein the sequence of chromosome is not changed, but due to some reasons, there are still some problems in the normal division or replication that is occurring, right? Then loss of proteostasis. Now, what is proteostasis? It is basically the maintenance of the normal proteins in our body, right? What is loss of proteostasis now? Due to genomic instability or maybe epigenetic alterations, it will not only affect the replication or division, it will also affect transcription, right? For example, the instance that I stated, a transcription factor might not be able to come and bind, right? So now, whatever mRNA had to be transcribed and whatever protein had to follow that will not happen, right? So that could lead to loss of proteostasis. Apart from this, there could be other kinds of things that are occurring, right? For example, post transcriptional modification might not work properly. Post-translational modifications might not work, due to which maybe the protein is being formed. The gene is functional, it is forming a functional mRNA, but it might happen that the functional mRNA is also forming the correct protein with respect to its sequence, right? But due to some problem with the chaperones that fold that protein, or some maybe one residue is altered, due to which the correct folding of this protein can't occur. And now the whole protein is useless, right? So it will either accumulate or it will be degraded, but we will not have that protein to perform the function that it had to perform. Right. Other kinds of hallmarks of aging that is written over here is antagonistic hallmarks, right? So, in response to damage, these things might occur, right? So, it could be cellular senescence, that is aging of cell and eventual, cellular arrest, right? Cell cycle arrest. Then mitochondrial dysfunction. Mitochondria is one of the most studied organelles at this point of time in the cell, right? Why? Because mitochondria is a powerhouse of the cell. That is what we have been taught since when we were in school, right? Now, these mitochondria, hello,
Ma'am, your sharing is not appearing now. It's gone.
yeah, I don't know what just happened. Just a moment, let me just try to share my screen again. I hope it is still going on. Yeah. Okay. It is visible to me. I hope it's visible to you again as well, right?
Yes, ma'am.
So
Yes.
we were talking about mitochondria actually. So mitochondria, as I said, is one of the most studied organelles that is as of today, right? So it is a powerhouse of the cell, and it also is one of the organelles that has its own DNA, right? And it has its own double membrane structure also. So it is a very, very special kind of organelle inside most of the cells, right? And dysfunction, any kind of dysfunction, all these the quenching of ROS, the effect of ROS, if they are present, it will mostly affect mitochondria, right? And the process of generation of ATP, which is also happening inside this mitochondria. So mitochondrial dysfunction takes place. Mitochondria in the first place is very, very sensitive to very small cellular changes, right? So it will change its phenotype. It will try to, um, make up for any kind of loss that the cell is facing. But if these things happen in a longer duration, right, they are not corrected, the other mechanisms that it is making up for are not corrected, it will become a chronic condition, right? In chronic conditions, mitochondrial dysfunction occurs, right? It cannot really make up for all the loss that the cell is occurring, and fission of mitochondria normally happen, right? They break. And if mitochondrial fission happens, it's usually followed by apoptosis, autophagy, one of the pathways in which the cell death will eventually occur, right? Then there's also deregulated nutrient sensing that also we had discussed in ma'am's lectures, right? For example, one of the examples of deregulated nutrient sensing that was discovered was via the mTOR signaling pathway, right? These things will also affect. The other two that are left come in the integrative hallmarks, that is, they manifest the aging phenotype, right? What will manifest aging phenotype? If there is exhaustion of stem cells. So, what will stem cell exhaustion do now? Basically, we do not have the pool of cells that can form any kind of cell, right? Or that can readily divide. So if that pool of cells is lost, now we don't have any, backup to rely on. If our cells that we currently have, they are dying, and there is no pool of cells from which this cell can be revived, right? So we will just face a loss of cell, eventual loss of cell, right? That is what is going to happen. It is, you can, analogous to this, you can expect that, for example, you have the staple diet in your in your home, maybe rice, right? So you have a continuous supply of rice, right? Because whenever rice is going to get over, you can go to the market and you can fetch more rice, right? For example, kind of a lockdown was to occur tomorrow, right? And this could be a more prolonged condition, not for a day or two. Such a lockdown had to occur that now the supply of rice is not there in your city, right? So you will use whatever is there with you, right? After you have exhausted that, you do not have any pool to rely on, or any backup or storage where you can go and where you can get more rice, right? When you exhaust the existing supply of rice, that will be it. So after that, what will happen? You will not have that anymore, right? So it will kind of exhaust. Then altered intercellular communication. Now, cell-cell communication is very, very important, especially in terms of cellular signaling, right? So cell signaling is something that is driving us altogether, right? It is driving each and every process that is happening in our body, right? If there is an altered intercellular communication, it might lead to a lot of unprecedented changes, which might not be good for us. Okay. So moving on to question number 3. The question is, pick the correct statement about the mTOR nutrient sensing pathway. A, it senses low energy states by detecting high NAD positive level. B, it senses high amino acid concentration. C, its down regulation or inhibition extends longevity in various organisms. Or D, its upregulation or activation extends longevity in various organisms. I would urge you to go through the options because, okay, let me just tell that to you. More than one of these options are actually correct. So let us just go through the answer. The correct statements regarding mTOR sensing pathways are that it senses high amino acid concentration, right? Among other nutrients, it also senses high amino acid concentration, and its down regulation or inhibition extends longevity in various organisms, right? So I have kept illustrations to explain both these things. So this was the one of the simplest illustrations that I could find. It is from a book chapter, I think, by me, Sopyun, from Nutrients. It was published in 2017. And in this, we see that there are two forms of mTOR. There is mTOR1, mTOR1 and mTOR2, right? And both of them will kind of be affected by various kinds of energy supplies in the body, right? The one that we are talking about right now is mtoc1, because that is directly sensing high levels of amino acids, right, like we talked about. So when there is higher level of amino acids, this mtoc1 will sense it, and it will basically promote cell growth, it will promote metabolism. mTOR2 similarly will promote cell survival and cytoskeletal rearrangement. And along with all this, mTOR1, when it senses high energy levels, oxygen levels, amino acid levels, etc., it will also stop autophagy. Right? So it is basically promoting cell growth and survival while it is stopping autophagy, right? But as you might remember from ma'am's lectures, autophagy is not bad for you, right? It is a programmed it might occur through various pathways, one of which might be cell death might occur through various pathways, one of which might be programmed also, right? And why is this programmed cell death important for you? Because the cell is willfully or willingly making this choice that yes, I think that my survival might be toxic for the surrounding cells or for this organism in general. So I think it is time for me to program or to undergo cell death, right? So it might not always be uh, uh, good that no cell death is occurring. So it has been seen. This is another illustration that I wanted to show you. So this is a representation or kind of a cartoon that is representing C. elegans, right? So C. elegans is one of the organisms that is widely studied. And in this particular organism, these group of authors had basically studied that in the when they normally studied mid age of C. elegans, they saw that in the somatic stem cells, the telomere length was like it was mediocre, right? It was not completely very short or very long, right? So this is representing the telomere length. Red is representing the maximal telomere length, and green will represent the minimal telomere length. Yellow will be telomere length lying in between, right? So please assume over here that at the beginning of life, the organism will have telomeres that are all red, right? As it progresses or as it ages, what will happen is that its telomere will keep shortening, right? Eventually, in the old age organism, you will see that the telomere length corresponds more to this green color, right? So, when the author studied that the C. elegans in the somatic stem cells, they normally showed this yellow colored or medial kind of uh, telomere length, right? And when these cells divided, right, after they had undergone multiple mitotic divisions, and when they had become a senescent cell, right, a post-mitotic cell or a senescent cell, and after they had quit dividing altogether, they could see that the telomere length was really less, right? It fell in this green area. But what they did was they experimentally starved this particular C. elegans, right? So when they saw that they took control population and the starvation population, right? In the control population, there was no starvation that was done, and in the other experimental set, the starvation was actually performed, right? And it was seen that in the control population, this kind of result was only seen that the somatic stem cells showed medial kind of telomere length when they were seen. But when starvation was actually performed, it was seen that the starved somatic stem cell retained its telomere length, right? And they checked multiple, downstream pathways or upstream pathways that might be responsible for this. And they found that mTOR was decreasing, right? When starvation was occurring, mTOR was decreasing. Okay, because nutrient sensing was not being done over here. Since then, there was not, a lot of nutrient that was present. So mTOR goes down when starvation occurs, and it basically retains the telomere length, right? After this particular study and other studies in this particular field, came up the hypothesis of intermittent fasting actually. So I would not suggest it to anybody, but what I would like to point out is that it is a very, very fascinating concept, and that concept is kind of built on these kinds of studies, right? And what it says is that if intermittent fasting is done, it will basically reduce the cellular aging process or cellular senescence process, right? It will decelerate this process, right? I'm so sorry. So I would urge you to go and read more about that. And this was what this particular paper talked about. You can read this paper also. The link has been provided. Yeah. So question number 4. Which of the following diseases is a consequence of too little apoptosis? Right? Is it autoimmune disease or Parkinson's disease or AIDS or stroke? So we have just talked in the previous slide that it is not important that if cell death is not occurring, it will be good, right? It might not be good in some cases. Just give me a little break, go through the question. Yeah. So the answer for this particular question is autoimmune diseases, right? So before I explain why this is, what is one other disease that was mentioned in ma'am's slides also, which is also a consequence of too little apoptosis? If anybody could just recall that. Okay, so let me tell that it is cancer. Okay, and cancer was talked about also in detail in the last session that was taken by ma'am. So why autoimmune diseases is due to too little apoptosis? Autoimmune diseases is when basically our own immune system is attacking our own cells, right? So something has gone wrong in our own body cells, either in our body cells or either in the recognition ability that is present in the immune cells, right? So at least at one point something has gone wrong, and the result of which is that the body's immune cells are attacking the body's own cells, right? And that is what is autoimmune diseases, and it is actually a consequence of too little apoptosis. So when these apoptosis is deregulated, in the sense that you know some kinds of cells, after they have accumulated certain uh, toxic proteins or certain level of ROS, etc., they undergo a programmed cell death, right? And most of the cells in our body, leaving a few, have this regenerative capacity in the sense that they can mitotically divide, right? So once this particular cell will die, there is the cells are mitotically active and they can cover up for its death, right? So it is a very, very natural process. But when this natural process will not occur, right, it will somehow stimulate some signaling pathways that okay, because it contains material that might be toxic, right? So it will somehow induce some kind of pathways due to which or which will alert our body's immune cells, right? Which could lead to autoimmune diseases. It could be one of the pathways, right? The other thing could be cancer. Why? Because now the cells are not dying, right? The cells that should have died are not dying, and that doesn't mean that the cells neighboring it will not be dividing, right? If they are mitotically active cells, they will keep on dividing, right? So this particular area will increase in the cell density, right? Because in cancer cells, as you might know, there is no contact inhibition also that is present. So normally in cells, what happens is that if there is a mitotically active population of cells, what will happen is that as these cells divide, the cells that were previously already present in this boundaries, if they will also receive a cue that okay, now new cell has arisen, right, in this particular territory, so maybe I can undergo apoptosis because this particular cell is healthier than I am, right? So that is contact inhibition, that it will not allow uh, too many cells be present in just right next to each other, right? Each cell needs certain amount of space to be present, right? And once something is mitotically active and it is dividing continuously, the older cells, they will die by apoptosis. So that doesn't happen in case of cancer, right? They lose this contact inhibition property and they keep on dividing. So they will form a mass of cells, right? Sometimes if it is on the, uh, surface, you will be able to see it as a growth or as a benign growth or tumor, right? So that happens in case of too little apoptosis. Now, in most of the neurodegenerative disorders, including Parkinson's disease, Alzheimer's disease, and some kinds of epilepsies, etc., what happens is that there is too much of apoptosis. That is why it is degenerative, right? So the neurons are dying, right? The neurons are one kind of cells that are not mitotically active. Only some neurons can divide, not all of them, right? So there is a very, very tiny niche of neuronal stem cells that is actually present. Most of the neurons, once formed, cannot be replaced or cannot mitotically divide, right? So if there is a lot of cell death that is occurring, or through too much of apoptosis is occurring in these cells, they will probably be detrimental for the organism, right? Similar is the case for AIDS and stroke. Okay. Question number 5. Which of the following proteins can physically go to mitochondria to promote the release of cytochrome C? Your options are A, BAX and BAC, B, BCL-XL, C, BAX and APAF1, or D, BCL2.
Option A, ma'am.
So it is actually option A, that is BAX and BAC, right? So these are two proteins that can go to the mitochondria, and this is a very good representation of this. That what happens is that this is a mitochondrial membrane, right? This is the outer mitochondrial membrane, and this is the cristae and the inner mitochondrial membrane, right? And within this mitochondrial membrane, in the intermembrane space, is present this protein that is cytochrome C, right? Which is marked over here in pink. Okay, so normally it is present in the intermembrane space inside the mitochondria. Now, what happens? There are some kinds of proteins like BAX and BAX or BAC, right? Which on receiving the apoptotic signal, they can go and they can sit on this outer mitochondrial membrane, right? And they can basically they have multiple kinds of domains, right? They out of which one domain, that is BH3 domain, will be exposed, and it will basically attract other back and backs particles, right? Other back or backs proteins. And now this BH3 domain that was exposed will bind to the groove of the other, this protein that has come, right? And similarly, it will recruit other such back or backs proteins, and these proteins together will, line on the outer mitochondrial membrane, and it will form a core, right? And this, sorry, a pore. And what will this pore do? It will basically create a channel through which this cytochrome C that is present inside the inner membrane space of this intermembrane space of this mitochondria can now pass out from this core, right? So there is another illustration that I have picked from Wikipedia, right? Over here, you can see that again, cytochrome C is present in the intermembrane space. And in the mitochondria, when the back or backs come, they form a complex, and this complex is termed as MAC, right? Mitochondrial apoptosis induced channel. Right, so this pore is kind of like a protein channel, right? And what this channel will do, it will aid the transport of cytochrome C out of the cell, right? And this can be prevented by the presence of BCL2. Okay, so this can be prevented by the presence of BCL2 over here. So I hope this particular part is clear. Okay. Now, among the following proteins, which protein inhibits apoptosis? BCL2, BCLXL, STAT1, and E2F2. Now, we have just discussed this, so I am not giving you much time to ponder upon this question, right? Uh, BCL2 is the correct answer for this one. In the previous illustration, also, I showed that this formation of this mitochondrial apoptosis induced channel that is occurring by the physical presence of back and Bax protein on the outer mitochondrial membrane can facilitate the transfer of cytochrome C out of the membrane, out of the mitochondria, right? And it can be stopped by the presence of BCL2. Right now, how does this happen? Uh, One second. This is how, right? So, when there is a normal condition in which apoptosis does not have to occur, right? And a cell survival signal is on, right? What will happen at that time? There will be survival factors, right, which will go and bind these backs and back proteins, right? These factors will be BCL family proteins like BCL2, BCLW, other BCL family proteins, right? And they will cause the inhibition of backs or back, right? Because they are bound to backs or back. So these backs or back can't bind to other, backs or back proteins to form that core complex, so that channel cannot be formed because they are bound by these BCL2 proteins in general, right? So once there is inhibition of backs or back, the cell continues to survive because the cytochrome C is inside the cell. But in case of apoptosis, through different pathways, it could happen that the death signal is obtained at the death receptor, right? Due to this, either through caspase 8 or through caspase 9, through different pathways, intrinsic or extrinsic pathway, right? It could happen that there are other proteins that are activated, right? When these other proteins are activated, they go and they attack BCL2, right? And once they attack BCL2, what will happen? They will remove BCL2 from this outer mitochondrial membrane. Right, once this BCL2 is removed from this outer mitochondrial membrane, because now it is bound to these cell survival withdrawal factors such as HRK, BIM, etc. Right, what will happen? Now back and backs can interact with one another, right? Because BCL2 and BCL2 family proteins are no longer binding it. They can interact with one another. They can form the complex on the outer mitochondrial membrane, and it can facilitate the export of cytochrome C out into the cell, right? And this can trigger this cytochrome C release can trigger apoptosis, right? So that is how apoptosis will occur, and that is how BCL2 actually prevents from apoptosis from occurring, right? If this survival factors are present, right? um, if the death signal is not received or there's no ligand on this particular receptor, the BCL2 will continue to remain bound to back or back such that they cannot form an oligomer among themselves and not cause the translocation of cytochrome C out of the mitochondria, right? So that is how this process occurs. Okay. So question number 7 is which of the following is a proto-oncogene? Right, your options are retinoblastoma RB, P53, BRCA1, c-MYC, all of the above or none of the above.
Option D, ma'am.
Which one?
Option D.
Yeah, so that is correct. D is actually the correct option, right? So the other ones are either tumor suppressor factors or oncogenes themselves, right? But which of these is a proto-oncogene? It is c-MYC, right? So as you might remember, what is a proto-oncogene? A proto-oncogene is a gene that has the potential to become an oncogene if it has some kind of a mutation that is occurring on this particular gene, right? So what is when it is a proto-oncogene, it usually has a suffix, no, sorry, a prefix followed by C, and when it becomes an oncogene, now this prefix changes to V or to nothing. The C is just removed, right? So what happens if there is a proto-oncogene and if mutations occur in these particular proto-oncogenes, right? They will deregulate cell division. That could mean that the cell division might go on continuously, it might not go at all. But in case of cancer, what happens? It will just go on infinitely, right? So there will be no more regulation of cell division that will occur, right? Whether the nutrients are there or they are not there, they will scavenge nutrients from other surrounding cells, but the cell division will go on, right? So these genes that are proto-oncogenes are denoted by three-letter names such as cis, Re, Mic, etc. Right? Since many of the oncogenes were first discovered as mutated cellular genes that were incorporated into nucleic acid of RNA tumor viruses, the activated forms are denoted as V-onc, as in V-cis, right? So oncogene might have the prefix V or V-Mic, for example, right? Or it might just be MYC, right? And the proto-oncogenes are C-onc and C-cis or C-MYC, right? As we just saw. Then we come to question number 8, which is in C. elegans, which of the following active proteins would prevent apoptosis? Is it SED4 or SED3 or SED9 or all of the above?
Option C, ma'am.
Yeah. So SED9 is actually the correct option. Uh, so I have kept a very simple representation over here as well, right? So if you first look at just half this figure, just let me zoom in. Yeah. If you just look at this part, right, where SED4 can activate SED3, which can in turn activate apoptosis, right? But SED9, if it is present, it will come and it will inhibit SED4. If SED4 is inhibited, it will not be able to activate SED3 and apoptosis will not occur, right? So that is the simple concept behind it. So what does this mean? Just a moment. Yeah, what does this mean? That other proteins, SED4 and SED3, right? If they are active, they will kind of uh, ex- uh, they will cause apoptosis to occur, right? But if SED9 is active, it will prevent apoptosis from occurring, right? Similarly, if EGL1 is active, it will also, what will it do? Will it prevent apoptosis or will it cause apoptosis if EGL1 is present? Okay, it is a simple question. It is just about reading the flow chart. Okay, so if EGL1 was present, it will mean that SED9 will get deregulated, right? So it will not be sorry, it will not be able to function, right? Because it is inhibited. And what was the function of SED9? It was to inhibit SED4. So if EGL1 is present, SED9 will not function. So SED4 will function normally, and it will activate SED3, which will activate apoptosis. So if EGL1 is active, apoptosis will occur. But if EGL1 is also not active, right, then there will be no inhibition to SED9. Right, SED9 will perform its function, that is, it will inhibit SED4. If SED4 is not activated, it will not activate SED3 and apoptosis will be prevented. Okay. Yeah. So question number 9. Pick the correct statement about cancer cells. A. Abnormal, unrestrained proliferation. B. Invasion of surrounding normal tissue. C. Cancer cells have somatic mutations. Or D. All of the above. Okay.
Option D, ma'am.
Yeah. So the correct answer over here is option D, right? Because cancer cells will have abnormal unrestrained proliferation, like we talked about. So they will not undergo cell death. They will keep on dividing, right? They will have a T-regulated cell cycle division. There will be no restriction, no contact inhibition, right? Because of all these things, it will invade the surrounding normal tissue also, because it will not have any inhibition, right, to grow. Whether it is growing in its own region or it is spreading to other regions, it will have no inhibition. And they will also have some kind of somatic mutations, because uh, if the cell cycle was working properly, if there was any kind of mutation, it will be arrested and it will go and check, the checkpoint will not be passed until that mutation is resolved, right? Mostly. But in cancer cells, what happens, since the cell cycle checkpoints are not active, right? Because the cell cycle is deregulated, now somatic mutations can accumulate in cancer cells. Okay. So the answer over here is all of the above. So this representation uh, has list has listed the different kinds of characteristics of cancer cells. One, there are 10 different kinds of characteristics that are mentioned. That is, they avoid immune destruction, right? So they will evade the immune system. They will enable replicative immortality. So as you might know, the cancer cell lines are immortal. That is, they can continuously be, passaged or continuously be grown in the cell culture dishes also, right? They do not follow the Hayflick limit. Then they activate invasion and metastasis. They induce angiogenesis, that is, they will induce, since the cells are now growing in number, they will want to have the nutrient sources. For that, they will induce the formation of blood vessels around this cancer, right? Then they would resist cell death. They would deregulate cellular energetics. They will sustain proliferative signaling. They will evade the growth suppressors. They possess genomic instability and mutation, like we talked about, and they mediate a tumor associated inflammatory response. Right? Okay. So, your final question for this particular segment is, pick the correct statement regarding caspase 3. Right? It causes DNA fragmentation by activating DNases. It dephosphorylates myosin regulatory light chain. It is activated by ROCK1 kinase, and it is the activated caspase 3 leads to membrane blebbing. So which out of these is the correct option? 1, 2, and 3, 1, 3, and 4, 2, 3, and 4, or all of the above? This is a slightly
Option
trickier
A.
question. Option?
A, ma'am.
No, actually, that is not the correct option. The correct one is option B. Okay. Because it will not dephosphorylate myosin regulatory light chain, but it will activate caspase 3. Active caspase 3 will lead to membrane blebbing. I thought these were the two easy ones, and you guys might be confused in 2 or 3, right? That whether it is activated by ROC1 kinase or not, or this one. But this fourth one is a given, right? This is a function of active caspase 3 that it will cause membrane blebbing, right? And which two options have this fourth one? It is B and C, right? Now from B and C, third option is also there in both of them, that it is activated by ROC1 kinase, right? Now, out of the first two, which one will be correct? The first one, that is caspase 3 activation, will cause DNA fragmentation by activating DNases, right? Because what will caspase 3 do? What is it a marker of? It is a marker of apoptosis, right? What is a function of apoptosis? Characteristic feature of apoptosis, that it will the chromatin or DNA fragmentation will happen in it, and membrane blebbing will happen, right? So 1 and 4 are definitely going to be there, right? This is the only option that has these two, right? So even if you did not know about this 2 or 3, you could have made it out based on the features of apoptosis. And if you know that caspase 3 is one of the effector molecules in apoptosis, right? So what happens in case of apoptosis, as we have already read, we can briefly revise it. There are two kinds of pathways that can lead to apoptotic cell death, right? One is the intrinsic pathway, in which there will be some kind of DNA damage or cell stress that will arise from within the cell, right? It can, you know, stimulate P53, which in turn can stimulate BAX. This BAX can physically go, and it can interact with other BAX molecules or BAC molecules on the surface of this outer mitochondrial membrane, and that will basically release cytochrome C, right? If such responses are present, they will inhibit BCL2, and thus this inhibition of cytosome C release will not occur, right? It will not occur, right? This cytochrome C that is released, right, along with procaspase 9 and APAF1, it will form a complex that is known as apoptosome, which will go and it will, activate this caspase 3, right? It will cleave the procaspase 3 in a way that it is activated into caspase 3, which is an effector molecule for apoptotic cell death. The other alternative pathway for this is the death receptor pathway, right? So there will be a death ligand, that is some extrinsic kind of cell signal that is signaling cell death, right? It will basically go and it will activate caspase 8, right, from the caspase, and this caspase 8 can also function similar to caspase 9, that is it can activate caspase 3, and that will again lead to apoptotic cell death, right? And what are the features of apoptotic cell death? The cell membrane will remain intact. There will be blebbing that will occur, that is finger-like processes will arise, right? The organelles will break down, but the unlike necrosis, cell membrane, they will be bound by cell membrane even when they are broken down and secreted as vesicles or removed as vesicles, which will be or phagocytosed by either neighboring cells or professional autophagosomes, right? Along with it, what will happen? There will be fragmentation of DNA and chromatin that will occur, right? So that is it for this particular segment. And we'll just quickly move on to the other part. Yeah, so for the people who have joined a little late, the second segment of this particular lecture is going to be some extra concepts. So this time I've not included questions. Actually, I forgot to remove this particular, you know, part. So in this particular extra concept session, what we'll talk about is where we had left from, right? So last class, if anybody of you attended, you might remember that we were speaking about chromosomal folding and how histones were working, right? What is the level of folding that is going on, etc. And we had discussed a few techniques also pertaining to this, um, topic. And now what we are going to do is that we will pick from where we had left off. So we had left off at lamp brush chromosomes. I told you that is something I really wanted to discuss, but I had not had the time to, prepare slides for that. And I wanted to discuss it at length. So we are going to discuss about lamp brush chromosomes, polytene chromosomes, these special kinds of chromosomes, interphase chromosomes that are present in various organisms that have actually indicated or given us hints as to what kind of chromosomal packaging might be going on in our own chromosomes as well, right? So that is what we'll be discussing in today's lecture. During the session itself, we discussed, I sufficiently, I think, about the aging cell, senescent cell, and aging and senescent cell. I also wanted to discuss about apoptosis, but again, that is not what we are going to do, at least in today's lecture, right? So, for now, we'll discuss the chromosomal packaging and chromosomal folding, and apoptosis and cancer are the two things that I will pick up in the following week's extra session, right? So, in that session, I have planned like that only, that I'll discuss apoptosis, necrosis, then other kinds of cell death, the newer versions that have come up, like ferroptosis, for example, right? So some other newer versions of cell deaths we can discuss over there. And along with that, we can talk about neurodegenerative diseases and cancer, right? So for now, we can begin with this particular session. Right. So this was actually the last slide that I had kept in the previous week's session, right? So it talked about non-histone proteins, which organize long chromatin loops, right? Because we had discussed some experiments also, wherein we had seen, and in ma'am's lectures also, you had seen that the professors that were invited speakers for that week's lecture had described how SMC proteins, various SMC proteins help in the formation of these DNA loops, etc. Right? So this was an image or an electrograph, electron micrograph image of the lamp brush chromosome that was one of the first such chromosomes that was to be seen, right? It shows a short segment of the lamp brush chromosome in the nucleus of an oocyte from the newt node of thalamus virescens, right? So this particular kind of chromosome is a interphase chromosome that is usually seen in amphibians, right? So we will discuss a little bit more about it. So why do we want to discuss about this at all, right? So let us begin with that, because I don't expect all of you to have been there in my previous class. Also, I don't expect all of you to have understood why we are discussing this. Okay, I think it got disconnected for me. Uh, Wait, I'll have to stop sharing. Share again. Yeah, I hope I'm there in the meeting at least. Yeah. So, okay, now that it has disconnected, let's just have a short water break. Okay. So I hope I'm visible and audible. I'll just share my screen again. Yeah. So what I was talking about was, I hope you did catch my previous slide. So this was the slide that we had left at in the previous session. That was what I was discussing. And what I was saying was that we were talking about very briefly, we talked about what these lamb brush chromosomes are and how they were the first chromosomes in which these loops were actually visible, which actually gave the idea to these researchers that that is how folding might occur in higher order organisms like us also, right? And that is what we discussed in the previous session, that there are some SMC proteins and some other non-histone proteins that are involved in the formation of such loops, and that is how folding is occurring in our chromosomes also, right? So just a moment. Received. Okay. Yeah. With that, before starting with what actually these Lambrush chromosomes are, I wanted to discuss again why we at all need to discuss what they are or look at them, right? So, what was the bigger question? The bigger question was to understand how chromosomal packaging is occurring, right? In the previous session, we saw that up to a certain limit, the packaging occurs because the DNA is folded around the chromatin thread, or the DNA is folded around histone proteins, and then these histone proteins also in a solenoid manner or in a double helix kind of manner, they further fold and form loops such that a 30 nanometer fiber is formed. But even after this 30 nanometer fiber is formed, right, which is the thicker version, not the 10 nanometer, which has beads on strings. But when those beads on strings are not actually visible because they are also arranged in the solenoid manner or the other manner, right? After that, the thicker thread that is formed is the 30 nanometer fiber, right? As a 30 nanometer fiber, a typical human chromosome would still be zero point 0.1 centimeter in length, right? Before this 30 nanometer fiber, also, we talked that the largest chromosome could be up to 10 centimeter in length, right? Even after this much compaction has been done, even as a 30 nanometer fiber, the human chromosome would still be zero point 0.1 meter in zero point 0.1 centimeter in length, right? Now this is still too big because our cells are in microns, right? And nucleus is even smaller than that. And this point, and after that, it will be able to span the nucleus more than 100 times, right? So that is actually true. Then there must be a still higher level of folding even in the interphase chromosomes, right? So we say that the chromosomes are most condensed when they are in the mitotic phase, right? That is when that solenoid structure and all also came actually effectively, right? But even in interphase, they will not just be lying around as a 30 nanometer fiber also. Why? Because they cannot be accommodated within the nucleus even in this particular form, right? So they must still possess a higher level of folding that we don't understand as yet, right? So how did we find about that? Although the molecular details are still largely a mystery, right? We don't exactly know what is happening. This higher order packaging almost certainly involves the folding of chromatin into a series of loops and coils. Now, there might be some, evidence that we have or some observation that we have made due to which this is what we think, that there might be a series of loops and coils due to which it is occurring, right? It has had to come from somewhere, right? It will not be that okay, I think this is going to be the case, but that thought is going to be based on some other observation maybe, right? So this chromatin packaging is fluid and it will frequently change in response to the needs of the cell, which also we are going to talk about today. Right. So when we talk about the lamp brush chromosome, right, which was the first image that I had shown you, uh, it has given us some insights to determine the global structure of chromosomes, right? So these very unusual lamp brush chromosomes, which are actually the largest chromosomes that are known, are paired in preparation for meiosis, right? They are clearly visible even in light microscope. So other chromosomes are not that easily visible, okay? But these are clearly visible even without staining under light microscope. When they are seen to be organized into a series of large chromatin loops emanating from a single chromosomal axis, right? So again, this is the same representation that I showed you earlier. These are what the Lambrus chromosomes look like. Okay. So when we talk about these chromosomes, what are the insights that they have given us? In these chromosomes, a given loop always contains the same DNA sequence that remains extended in the same manner as the oocyte grows, right? So if we see this particular loop that is extended over here, right? Even as a oocyte will grow, the same amount of the same DNA sequence will be present in this loop, right? It will not continually change. Okay, these chromosomes are producing large amounts of RNA for oocyte, and most of the genes present in the DNA loops are being actively expressed, right? So what is the purpose of RNA production? It means that they are transcriptionally active. These chromosomes have genes that are transcriptionally active, right? That is why it is able to produce large amounts of RNA. And most of the genes that are present in these DNA loops are actually being actively expressed. So that is what it means, right? The majority of the DNA, however, is not in loops but remains highly condensed on the chromosomal axis, right? So you can see that there is this one big loop that is not really in this picture, right? Then there is this other big loop, this third very big loop, then this fourth big loop. But most of the DNA you can see is still highly condensed over here, right? Most of it is not in these loops, right? So that is what this statement says. Majority of DNA is not in loops but remains highly condensed on the chromosomal axis, which is this, where genes are generally not expressed, right? So whatever genes will be transcriptionally active or actively being uh, uh, replicated or transcribed, that will probably be present in the loops that are extending outwards, and not in the chromosomal axis where they are densely packed with one another, right? This is another, representation of the Lambrus chromosome. So if you see in an electron micrograph, this is what it looks like, right? If it is not stained, this is what it looks like. Over here, you see that there is a dense chromosomal axis in between, but there are multiple loops that arise from this, and all these processes that you are seeing apart from the dense central axis are all loops that are arising, and all these loops are transcriptionally active genes or contain transcriptionally active genes, right? And over here you can see that they are fluorescently labeled, and you can clearly see these RNA forming loops, right? The loops that are responsible for more RNAs that are being formed, right? And around that, you can see that these are uridine, particles that are labeled because they are precursors for RNA. So, from the study of this lamp brush chromosome, the researchers came up with a model for the chromatin domains, ठीक है, which is a inner lamp brush chromosome. So this model, they later said, would probably be applicable even in other organisms in which they cannot actually see such loops or visibly, you know, photograph such loops because the chromosomes are not that big, right? But it might be the same process that might be going on in other chromosomes as well, right? So what was the model? In this model, they said that there are regions which are highly condensed in the chromatin, there are less condensed regions in the chromatin, and there are extended chromatins in looped domain, right? These extended chromatins are actually the least condensed regions, and this is where most of the transcription and replication is taking place, right? Okay. The set of Lamprecht chromosomes in many amphibians contain a total of about 10,000 loops resembling those that are shown over here. So these are the loop regions, right? Around 10,000 such loops are present in the amphibians. in the Lamb-Rush chromosomes of the amphibians. The rest of the DNA in each chromosome remains highly condensed, which we can see over here. 4 copies of each loop are present in the cell since each Lamb-Rush chromosome consists of 2 aligned sets of paired chromosomes, paired chromatids, right? So, if you might remember, there are homologous chromosomes, right? Homologous chromosomes have each chromosome in that pair have 2 chromatids, right? Yeah. So this is one part of a homologous chromosome. This is one chromosome, sorry, and this is the other chromosome. Now, these 2 chromosomes are homologous to one another, right? So they are homologous chromosomes, but they have 4 chromatids, right? And each chromatid will have its own loops that are present, right? So each identical domain will be present in 4 copies actually, right? So 4 copies of each loop are present in the cell since each Lamb-Rush chromosome consists of 2 aligned sets of paired chromatids. I hope this statement makes sense to you. Okay. This 4-stranded structure is characteristic of the stage of development of oocyte, which has arrested at the diploidine stage of meiosis. Okay. Uh, it is thought that the interphase chromosomes of all the eukaryotes are similarly arranged in loops. Okay, so it is not just in these Lamb-Rush chromosomes, but in all, it is thought that other interphase chromosomes of other organisms will also be similarly arranged in loops. Okay, although these loops are normally too small and fragile to be easily observed in a light microscope. But there are other methods that can be used to infer their presence, right? What are these methods? There are, please give me a moment. I'm just getting a call. Yeah, I'm sorry for the interruption. Okay. Yeah, so where was I? I was on this particular point that modern DNA technologies have actually made it possible to assess the frequency with which the 2 loci along an interphase chromosomes are held together, right? So, for example, over here, this and this loci will be held together, right? Not this and this loci. So, we will understand that since these two are not held together and these two are held together and they are far apart, right? So it is possible that it is forming a loop at this location, right? But over here, these two are close by. Even when we talk in the sequence, they are close by. So we will understand that, okay, they are not forming a loop. They are in that highly condensed region. It is present, right? Thus revealing likely candidates for sites on chromatin that form the basis of the loop structure. Okay. So, one such technology is the chromosome conformation capture method, right? Similar to this technology, and actually, a lot of improvements on this technology have been discussed with you in week 6 lectures by your professors, right? In this case, what happens is that there are DNA binding proteins that are cross-linked, right? They are treated with formaldehyde, and then cross links are formed, right? After they are cross linked, they are cut with restriction enzymes, right? Once they are cut with restriction enzymes, it will basically break your DNA into multiple fragments, right? At all these restriction sites that it recognizes. After that, we ligate the DNA. After that, these this particular fragments that are formed, they in this particular kind of mixture is diluted. Why is it diluted? Because once it will be diluted, then these fragments will move far apart, right? It will move a little farther apart, and after that, when we do ligation, only those fragments will be bound to one another which are most closely packed or which are cross-linked over here, right? Because of our treatment, the ones that are cross-linked will be ligated, right? Because if we add ligase over here, it could happen that this fragment goes and ligates over here, or this one goes and ligates over here, right? Then we will not assess this properly, right? So we dilute this mixture. After that, we ligate it such that the fragments that are close by get ligated. And after that, we remove the cross link, right? By heat treatment and proteolysis, because this is a protein that is present. After that, we will use the DNA probes, right? And we will test for the joint segments by multiple PCRs, multiple rounds of PCRs, right? So the DNA product is obtained only if proteins hold the two DNA sequences close together in the cell, right? Because we have cross-linked and undergone uh, uh, the restriction by nucleases, after which we have diluted it, and only then we have ligated it. So this is one of the methods for determining the position of loops and interface chromosomes, right? Because it will actually link the close by DNA regions. Uh, then these experiments and others actually suggest that the DNA in human chromosomes is likely to be organized into loops of various lengths, right? A typical loop might contain between 50,000 to 2 lakh nucleotide pairs of DNA, although there are loops of 1 million nucleotide pairs that have also been suggested. The chromatin in each individual loop is further condensed through poorly understood folding processes that are reversed when cell requires direct access to DNA packaged in the loop. Okay, so for example, there is a fold chromatin fiber that is present over here, right? There are multiple loop domains that are present. The proteins that form the chromosomes ke fold are shown over here. And through histone modifying enzymes, chromatin remodeling complexes, and RNA polymerases, what happens is that whatever region of DNA requires to be accessed directly, as it is mentioned over here, that will form a uh, loop, right? So this is a high level expression of genes in this loop, right? These will be low level expression of genes because over here maybe the transcription machinery or replication machinery will not be that easily accessible, right? Over here it will be very readily accessible because there's a lot of space for it. Okay, then we come to the other kind of chromosome that I wanted to discuss, which is the polytene chromosome, right? So again, for the global structure of chromosomes, there are insights that we have received from polytene chromosomes. These are basically chromosomes that are present in Drosophila salivary glands, right? This is a representation of the entire set of polytene chromosomes in the Drosophila salivary cell, right? So Drosophila actually have only 4 chromosomes, right? This is what a polytene chromosome looks like, right? Where these 4 chromosomes are very close by, and this is actually exactly what it looks like. Okay, we will understand what we have learned from this particular structure of polytene chromosome. Then some types of cells in many organisms grow abnormally large through multiple cycles of synthesis without cell division, right? So in this particular polytene chromosome, it is very, very huge, right? It is a special kind of chromosome only in the salivary glands of Drosophila. What happens over here, that cell division is not actually happening, but there's a lot of DNA synthesis that is happening, right? So what happens, that such cells in which this phenomena is occurring contain increased numbers of standard chromosomes, which are said to be polyploid. Okay, in the salivary glands of fly larvae, this process is taken to an extreme degree, creating huge cells that contain hundreds or thousands of copies of the genome. All the copies of each chromosomes are aligned side by side in exact register, like drinking straws in a box to create a giant polytene chromosome, right? So this is just an introduction about this chromosome. Now, what are the insights that they give, right? When the polytene chromosomes from a fly's salivary glands are viewed in the light microscope, you can see a distinct alternating dark bands and light interbands, right? Over here, you can see that there is black, white, black, white, black, white kind of regions that are present. So the black region is the dark bands and the whitish region is the light interbands, right? They are very well visible when we see these chromosomes under light microscope, right? And uh, these are visible and each is formed from 1,000 identical DNA sequences arranged side by side in register, right? About 95% of DNA in polytene chromosomes is in these dark colored bands and only about 5% is in these interbands, that is light in color, right? So why is it dark in color? Because the DNA is condensed and very close by. And why is the other one light in color? Because it is very, very spaced apart, right? So it is not showing anything over there. This is what it actually looks like, the bands and interbands, right? So when we look at it, this white colored region is not exactly, empty. It is just less dense. In this magnified version, you can see that. And the dark colored region are very, very highly dense. That is why this is what it looks like, right? So this is a micrograph of polytene chromosome from the Drosophila salivary glands. Now, what did we learn from these chromosomes, right? 1 second. Yeah. So a very thin band can contain 3,000 nucleotides. Like for example, this is one of the thin bands, or this is one of the thin band, right? It will still contain around 3,000 nucleotide pairs. While a thick band like this one will contain maybe 2 lakh nucleotide pairs in each of its chromatin strands, right? The chromatin in each band appears dark because of the DNA is more condensed than the DNA in interbands, and it may also contain a higher concentration of proteins, right? Because we know that in chromatin formation, it is not just DNA that is involved. There are multiple kinds of proteins that are involved in this formation, right? So protein density might also be higher in this band region. Okay. This banding pattern seems to reflect the same sort of organization that is detected in amphibian lamb-brush chromosome, which was described earlier. So it is also another, cue to us that, yeah, that whatever we are trying to extrapolate, that yes, this is what we have seen in the lamb-brush chromosomes in the amphibians, and we think this is what might happen in other organisms also. And in Drosophila also, we think that, yeah, this might happen and yeah, we can see this. This is a very similar pattern, right? Now, when we speak about polytene chromosomes, there are approximately 8,700 bands and 3,700 interbands. So the number of bands and interbands are almost fairly constant and fairly equal in the complete set of Drosophila polytene chromosomes, right? But the bands can be recognized by the different thicknesses and spacings, and each one has been given a number to generate a chromosome map that has been indexed to finish the chromosome index to the finished genome sequence of this fly. So I will show you an example of that. Have I not kept it? I have not. Okay. what were the insights that we take from these polytene chromosomes, right? Apart from this banding and interbanding pattern. The Drosophila polytene chromosome provides a good starting point for examining how chromatin is organized on a large scale, right? Previously, we saw that there are many forms of chromatin, each of which contain nucleosomes within different combination of modified histones. Specific set of histone proteins assemble on these nucleosomes to affect biological functions in different ways, right? So there are different kinds of histone and non-histone proteins also that are present, right? And what kinds of these histone proteins are present together or non-histone proteins are present together will actually determine what kind of function is being affected, right? So the recruitment of some of these non-histone proteins can spread for long distances along the DNA, imparting a similar chromatin structure to broad tracts of genome. Right? Such regions where all of the chromatin has similar structure are separated from the neighboring domains by barrier proteins. At low resolution, the interface chromosome can therefore be considered as a mosaic of chromatin structures, each containing particular nucleosome modifications associated with a particular set of non-histone proteins, right? So we can consider that why are these bands, sometimes they are very dense, sometimes the bands are also thinner, sometimes they are very thick, because there are similar kinds of histones and non-histone proteins that are also present in these thicker bands, right? In the thinner ones there are similar ones that are present, but they might have another function, right? The polytene chromosomes allow us to see details of this mosaic chromosome in light microscope, as well as to observe some of the changes associated with the gene expression. Okay, just a moment. Yeah. Okay, then there is another feature of these polytene chromosomes, right? Over here, when an insect progresses from one developmental stage to another. So what are the developmental stages of insects or Drosophila at that? So there will be the egg, there will be the larva, the pupa, and then the adult, right? So these are the different developmental stages. And as the insect will progress from maybe the egg to the larva or the larva to the pupa, there will be distinctive chromosome puffs that will arise, and the old puffs recede in its polytene chromosomes as new genes become expressed and old genes are turned off, right? So across developmental stages, we have, this knowledge that different kinds of genes will be active, right? Maybe that is what is even driving the developmental stages, right? Different genes being active, right? So when different genes have to be active, the loops have to kind of be modified, right? So what is this chromosomal puff? Right over here, you can see that this was a chromosome that was going, and in between, you can see that there is this puff-like structure, right? What is this puff-like structure? It is basically depicting RNA synthesis, because what is labeled over here, it is uh, a form of a 3H uridine. It is a form of uridine, which is a precursor for RNA synthesis, right? So this is determining that a lot of RNA synthesis means a lot of transcription is going on over here, right? And this puff will shift from developmental stage to developmental stage. Okay, because different kinds of genes have to be expressed in different developmental stages. Also, from inspection of each puff, when it is relatively small and then the banding pattern is still discernible, right? It seems that most puffs arise from decondensation of a single chromosome band. So over here, you can see that the rest of the banding pattern is discernible, right? And over here also, you can see that this is the light band area still, right? So over here might have been the dark band. So this basically indicates that one band basically decondense to form a puff, right? That is the indication that we could make from this observation, the inference that we could make from this observation, right? When we talk about these chromosome loops, the individual chromatin fibers that make up a puff can be visualized with an electron microscope, right? In favorable cases, loops are seen much like those that are observed in amphibian lamb-brush chromosome. So we said that in amphibian lamb-brush chromosome, since that is the largest existing chromosome, we can actually visibly see these loops. But sometimes, since polytene chromosomes are also fairly large, right? Sometimes in electron microscopy, we can actually see the loops over here also, right? And we saw, you might have seen in your lectures that now researchers have come up with techniques wherein they can sequester DNA from two ends, and they can, flow buffer in one direction so as to reveal these chromosomes even in human or other models, right? So when genes in the loop are not expressed, the loop assumes a thickened structure. Okay, possibly that of a folded 30 nanometer fiber, but when expression is occurring, the loop becomes more extended. Now, this is something we have already talked about. The chromatin located on either side of the decondensed loop appears considerably more compact, suggesting that the loop constitutes a distinct functional domain of chromatin structure. Okay. Also, in chromatin loops, there have been observations in the human cells, which also suggest that highly folded loops of chromatin expand to occupy an increased volume when a gene within them is expressed. Right? Example, a quiescent chromosome region from zero point 0.4 to 2 million nucleotide pairs in length appear as compact dots in an interface nucleus when visualized by fluorescence microscopy. However, the same DNA is seen to occupy a larger territory when the genes are expressed, right, with elongated punctate structures replacing the original dot. Right. So if you study the same chromosome region or same gene over different stages in the cell cycle, you can see that at some stage when the chromosome is very, very condensed, you will see it as a dot. But in other stages, for example, in the synthesis phase, when the chromosome is dividing, right, or it is being replicated, at that time you will see that it is not a dot anymore. It is elongated, right, because actively it is being replicated. Then we talk about chromosome arrangement. So you look at this figure. I will explain you in a moment what this particular figure is. Right. So there have come up new ways of visualizing individual chromosomes, which have actually shown that each of the 46 interphase chromosomes in a human cell tend to occupy its own discrete territory. Right. So these are different colors represent different chromosomes, right? And you can see that they kind of have their own territory, right? uh, which is within the nucleus. That is, the chromosomes are not extensively entangled in one another, right? So over here, what does this kind of staining represent? Over here, what is the staining that is done in the first place? Over here, there was a mixture of probes that was used, right? And each of these probes stains some particular part on some particular chromosome, right? So maybe I picked a couple of probes from one, and I labeled them with gray fluorophore, right? And then I picked a couple of them from a probe from chromosome 3, and I labeled them with some other color fluorophore, right? And then I'm representing all of them, right? So this is a simultaneous visualization of the chromosome territories for all of the human chromosomes in a single interface nucleus. Now, I would like you to look at this very carefully and make a very nice observation over here, that you can see that the homologous chromosomes don't necessarily lie together, right? For example, this is chromosome 9, and this is the other homologous chromosome 9, right? For example, we can find other ones. This is chromosome 17, and this is the other chromosome 17, right? Uh, then there are multiple other such chromosomes, right? So they don't necessarily lie together. That is what I want you to know, right? So they have been listed as different numbers: chromosome 3 over here and 3 over here, 8 over here, 8 over here, right? So that also I wanted to point out. So during interface, when it is not necessary that the chromosomes, homologous chromosomes will actually lie together. Okay. Also, The pictures, as these present only an average view of the DNA in each chromosome, right? Because we are choosing some probes. Those probes might also be at different locations, right? It will not indicate the location of the entire chromosome, right? Experiments that specifically localize the heterochromatic regions of a chromosome reveal that they are often closely associated with nuclear lamina, regardless of the chromosome examined. So, for example, over here in the middle, we are seeing chromosome 6 is present, right? But if we examine it properly, any of these chromosomes will somehow be associated with the nuclear lamina, right? Any of these chromosomes. So, DNA probes that preferentially stain gene-rich regions of human chromosomes produce a striking picture of interface nucleus that presumably reflects different average positions for active and inactive genes, right? Over here, maybe only the active genes are being represented. Maybe there is no representation at all for inactive genes. Okay. Uh, yeah. So, based on that, there was also a fractal globule model for interface chromosome that was proposed, right? Over here, you can see that in a three-dimensional structure, sorry, if you see in a three-dimensional structure, whenever a chromosome folding occurs, right, you can see that without knotting, there is loop formation and there is folding of chromosome or compaction of chromosome, but knots don't occur, right? It is just folding around one another without creating any kind of knots, right? So that is what is a fractal globule model of interfaced chromatin. Uh, Also, the chromatin can move to specific sites within the nucleus to alter gene expression. Now, I have a very good visual representation of this. But before that, let us go through what's written. So, a variety of different types of experiments has led to the conclusion that the position of a gene in the interior of the nucleus changes when it becomes highly expressed. What does this mean? That maybe when a gene was inactive, it was located at some particular region in a nucleus, but once it becomes activated, now it is present in some other region altogether, right? Thus, a region that becomes very actively transcribed is sometimes found to extend out of its chromosome territory, right? So we marked chromosome territories through those probes that we used, right? But if some region is very transcriptionally active, it will extend out of its chromosome territory as if in an extended loop, right? So the extended loop would look like what was shown in this fractal region also, right? Now, this was the representation that I was talking about. Over here, you can see that the chromatin can actually move to specific sites within the nucleus to alter gene expression. Over here, if I zoom it a little, you can clearly see maybe that, yeah, the red dot is representing the gene that we have marked, and the green is the entire chromosome, right? And you can see that when the chromosome is inactive, it is at the periphery, right? The gene is also at the periphery and the chromosome is also at the periphery. But when the gene is expressing, you can see that the gene is really, far inside the nucleus, and the chromosome has also detached a little from the periphery. From here, it has not detached that much, but from here, it has detached substantially, right? And you can see that how could it be possible that the gene is like so far inside while the chromosome is left over here? It could not be that the gene has broken off the chromosome, it has gone inside. After it will transcribe, it will come back. No. So what could possibly have occurred is represented in this image, right? So when the gene was off or it was not active, the homologous chromosomes that were detected by a hybridization technique, and the gene was marked, and it was seen that both of them occupied this periphery region, and they were, close to the nuclear lamina, right, or the nuclear envelope. And when the gene was transcriptionally active, it was seen that the chromosomes kind of detached a little from the nuclear lamina, and they might have formed an extended loop, right? Due to which these genes were visible right in the middle of the nucleus, right, and far away from the individual chromosomes, right? That could have occurred because of formation of loops, right? So it is yet another example that yes, uh, this might be the phenomena that is taking place, loop formation, right? For genes to be active, and that is how chromatin is actually being packed on a, higher level. Yeah, so that is it for this session from my end. I'll just let me just come back to the main screen and stop sharing it. So that is what I actually wanted to discuss, and I ran out of time last session, and I think you could understand why, because it was a fairly long topic to be discussed, right? So as I told you in the beginning of today's session also, that this is what we'll be discussing today. We are done with our discussion pertaining to that particular topic. Now, what I also did mention was that the different kinds of cell death mechanisms, uh, and what are the characteristic features of each one of them, and maybe even the cellular markers that are normally used in research laboratories to figure out which kind of a cell death process is going on, or whether it is going on or not, right? So that is something that we will discuss in the next session. So I'll prepare my slides accordingly. And that is it from my end. If you guys have any question, you may please go ahead and ask. Otherwise, I'll just stop the recording and end the session. Okay, so that is what I'm going to do. I'll just stop the recording.